Hello everyone and warm welcome to another webinar organized by ANT Neuro. My name is Farnoosh, I'm the head of education at ANT and I'm very happy to be hosting today's session on the principles and best practices of EEG hyperscanning. It's my pleasure to introduce our presenter today, who is my colleague Krittika Choudhury, who is currently the head of application support for ANT Neuro. She holds a master's degree in biomedical engineering from the University of Twente, the Netherlands, where she specialized in the neural motor systems track. After her presentation, there will be a video to walk you through a hyperscanning setup in action, and Dr. Fabio Barolo from the application team will provide you with some more details. He will also join the discussion together with Dr. Ishwar Gumar as the product manager for the research solutions to answer all your questions. So if you have any questions in the meantime and during the presentation, please feel free to write them in the questions chat box and we'll make sure to cover them during the Q&A as far as the timing allows. Also, there will be some poll questions seeking your active participation as the results will be shown live. So, Krittika, thank you again for taking the lead on this, and we're looking forward to learning more from you about the hyperscanning, which is a hot topic. We hear a lot, actually, in cognitive neuroscience societies these days. The stage is yours.
Thanks a lot, Farnoosh. Just to bring in a little bit of how this webinar is different from the other ones, I think this is one of the first ones that we as an application team want to introduce some concepts in the field to the audience. And the concept, like Farnoosh just said right now, that we want to share with you is hyperscanning, because it's one of the most emerging topics in neuroscience research. I will be joined, like Farnoosh said, later by my colleagues Fabio and Ishwar for the discussion round. I do have a short agenda covering the discussion structure that I want to follow today. I will start with a little bit of what hyperscanning truly is, a little bit on the timeline and distribution of hyperscanning studies up until 2019 or 20, some application use cases of hyperscanning. And for each of these settings, we would go over a schematic overview of the setup, as well as the hardware components that you would need to achieve this from an eego perspective. I will touch upon a little bit on the technical configuration that you would need to enable on the software side. I think it would be nice to also share with you some use cases where the eego system was deployed out in the field. Finally, I have a little bit of a review on existing hyperscanning methodologies and to leave you with freely available toolboxes and data sets for hyperscanning that are out there. As is the case with any sort of study that you want to do, there's also some challenges that are peculiar to the field, which I will cover in my slides as well. Finally, we have a product demo video by my colleague Fabio, which we would like to highlight at the end of my presentation. And as per always, we will have a Q&A session in the end. Without further ado, I go into my presentation. The first slide actually is a poll question, and I want to just understand the kind of audience we'll be interacting with today. So my first poll question is, are you familiar with hyperscanning studies in your field of research? I think the poll was just put up by Farnoosh. You have a couple of options, so yes, no, heard of it, want to employ it, or heard of it, a bit wary of it. You would have typically about 30 seconds for the poll. Farnoosh, could you let me know when the polls are in and closed?
Sure, let's give it some more seconds and we can close the poll. Can you see the results, Krittika, or should I read them out to you?
I think I see the results. So if I'm correct, about 45% say no, they've actually never heard of hyperscanning as a concept. And to those, I hope this webinar gives you a little bit of insight into what you can achieve with hyperscanning studies. About 27% say yes. So I'm glad to have some of you in the audience who already are familiar with such setups. And 23% say heard of it, want to employ it. So hopefully you're still interested in the rest of the presentation, where I hope to give you a little bit of an overview of such setups and studies. Without further ado, I will then get into my presentation. First being, what is hyperscanning? So hyperscanning by definition is when you're doing simultaneous recordings from two or more subjects. There is a variety of neuroimaging methods that have been employed for hyperscanning studies, right? So fMRI being one of them, EEG being one of them, FNER studies are really popular in this domain. And with all of these neuroimaging methods, the primary objective is to gain insight on topics such as social synchrony, on what cooperation versus competition dynamics look like, how your audience and a performer interact with each other, for example, and to lend a little bit of insight into team chemistry, performance, team mental models, and so. So all of these objectives are possible to answer or address with the hyperscanning study. This brings me to my next slide, which is basically what kind of motivation do you need for such hyperscanning setups? And I think that is most easily explained by citing literature. So here I have 4 examples. The first one that you see here is dealing with the tiger problem, which is popular in the fields of AI. But here they have adapted it to an EEG hyperscanning setup in context. You have 2 players, player X and Y, who have a reward in the form of a pot of gold behind a closed door, or they have a punishment in the form of a tiger behind another door. And both participants have to make use of audio that is provided to them. There's a little bit of task training that is provided to them to decide behind which door is the correct answer, and behind which door the correct answer being the reward in this case lies. Again, they're looking into 2 sorts of dynamics. So one is the cooperation dynamic, where both participants have to basically cooperate with each other to find the reward or the pot of gold behind the right door. And the second is the competition dynamic, where both participants have to compete with each other, and the first person to locate the pot of gold wins. This is one of the setups where hyperscanning could be employed. There is another one, which you see below. Here you have a response time measurement between 2 participants. What you see in the first slide is this gray marker, which tells the participants that the experiment paradigm is ready to begin. You see the green marker or the green slide with the green dot, which basically is the go signal for the participants. Once again, and here there was actually no EEG data collected, but more response time measures that were looked into. You have 2 participants, A and B. So for participant 1, you actually see the solid line. For participant 2, you see the dotted line in the picture. And when they cooperate with each other, they are supposed to work together to bring their response times more or less in the similar range. When they compete with each other, the person with the shortest response time wins. And what this study actually showed us is in line with what was found out as an outcome of EEG hyperscanning studies. But if you look at diagram C, so this particular plot over here, you see that the phase coherence is consistent, or the phase difference is consistent over time for the cooperation kind of studies, which is denoted by the blue color. And the phase coherence levels are not consistent over time for these competition kind of studies. You also see this in the polar plots that you see here. If you look a little bit towards the circle and the points over here, you see that when the participants are actually cooperating with each other, all of the points are clustered in more or less the same area or the same zone. The black line that you see gives you a single value of phase coherence between the 2 subjects. Whereas in the competition kind of scenario, you see that the points are more clustered over space in the polar plot. And there is a very, very less, so there is not a single value that exists that really sort of gives you an insight into the phase coherence between the 2 subjects. It's very distributed over time. The third kind of example scenario where a hyperscanning study could also be relevant is what you see on the top right now. So Hold on, I think I'm okay. You should be able now to see my screen again. You see a parent and a child listening to stories together. There are two kinds of inputs that are being recorded from both parents and children. So there's a camera input and there is a mic input. And of course, as we know that parent-child dynamics have a lot to do when it comes to fields such as emotional regulation. This is a kind of hyperscanning study. This is a kind of study that would be possible or easier done with a hyperscanning setup. Finally, I have here an FNER study, actually. So this is not EEG again, where they employ FNERs and GSR as two measures to look into the autonomic nervous system. So that's the part of the nervous system that is responsible for involuntary functions. The final outcome of this study actually goes into showing that inter-brain synchrony is more dependent on social interactions, especially when it comes to cooperation, rather than other inputs such as GSR level of increase or so. The GSR levels were found to increase as the task difficulty level increases. So the four pictures that you see here gives you an overview of the kind of setups that could be motivated by hyperscanning studies. I do have a little bit of an overview of the different kind of studies that do exist in literature. So like I said, first, it would give you an idea between task difficulty level, team coordination level, and mental workload in an interactive environment. It would give you, when it comes to diets, it would give you an idea on decision making that is employed during a competitive or cooperative task. This is also very helpful in the clinical realm, where, for example, age-matched, gender-matched controls and subjects can be looked into, and how there's deviations between a control population and the actual subjects. There was actually a study done for high-performing individuals or children in the autism spectrum, and controls were regular subjects who were gender-matched and age-matched with them. Finally, mother-infant diets or mother-child diets are also being investigated very much in the fields. Like I said, between the relationship that parents and infants share really is fundamental to how a child develops. And this is something that is also very explorable with the hyperscanning setup. This gives you a little bit of an overview of what motivates hyperscanning studies. But in terms of how hyperscanning as a term was originated, the first time this was used, hyperscan was the term that was used by Montag et al. In the 2002 paper. In that paper, they were looking at simultaneous fMRI acquisitions from two subjects. And these two subjects were playing a children's game called Handy Dandy. So one subject typically would hold an object in their hand, and the other subject would have to decide or find out which hand actually held the object. Now, while this is the case for the term hyperscan or hyperscanning itself, there are dyadic observational studies that existed in the animal population, in rhesus monkeys, etc., that predates this term. And this has existed in the last 40 years or so. So it's not new. Only the term is relatively new. I have here a list of keywords that might be relevant when you're looking at such studies. So it's not just hyperscanning, but it's also dyads, dual recordings, dyadic recordings that could help you get closer to the kind of studies that exist in the field. Why did hyperscanning as a concept gain popularity? It's because there was finally an option to look at neurocorrelates of social interaction. And to find correlations between subjects' data, you could do a final analysis and you look at phase coherence or connectivity, but you don't really need biomarkers of fiducials to find this correlation. Finally, now the state-of-art technology as we have it could solve a lot of hardware and software limitations of the past. So now is sort of like a good time to explore hyperscanning as a concept, because really sophisticated, complex, synchronized structures are possible with the kind of hardware that we have. This brings me to my poll question number 2, because before we go into the distribution of such studies in the field, I would like to know what you think. Which neuroimaging method do you think is most popular for hyperscanning? Your options are 4, so EEG, MEG, FNERs, fMRI, or none of the above. As always, you would have a couple of seconds to answer the poll, and I think the poll is now running. We'll most likely give it a couple of seconds. Parnush, please let me know when you've closed the poll.
Yes, around 70% have voted, so I think we can close it. And here are the results.
Thank you. I do see the results myself. So 59% of you said EEG/MEG is the most adopted neuroimaging method in these studies, and you are right. I will give a little bit more detail on this in the upcoming slide, but indeed 47% of the studies that exist for hyperscanning have employed EEG or MEG as the neuroimaging method. The least favored method was STAX, which was used for about 1% of the studies. I will go to the next set of slides, which covers a little bit on the distribution of these studies and application areas that you see hyperscanning studies being composed of. If the poll is closed, I will now start sharing the next screen. Yeah, so here what you see is taken from one of the review articles by Nam et al., which covers all of the hyperscanning studies that have existed ever since the term was coined in 2002 up until 2019. The paper itself was published in 2020, and of course, in the past 2 years, there have been more progress or more of an increase in the kind of hyperscanning studies that exist out there. As you can see, the first coherence-based fMRI study was done by Montaigne et al., where the subjects played the handy-dandy game. But first correlation-based fMRI study, where they looked into pairs of subjects, was actually done by Krittika et al. If you look at the studies that exist, so PC and PLV-based EEG studies, these are two analysis methods that have been used in the field for EEG. So PDC going to partial directed coherence, and PLV looking at phase locking values. So these were the two analysis methods that were adopted for these EEG-based hyperscanning studies. There's also functional connectivity that was explored a lot when you're looking at inter-brain synchrony between subjects. Finally, the first simultaneous EEG-MEG study was actually done in 2018 by An et al. Recently and lately, they have been exploring music a lot for hyperscanning studies also. So either music or looking at moments related to music recognition and listening for stroke rehabilitation, for example, or like you see here, the first EEG study was actually done by Greco et al. In 2018. Ever since then, there have been quite some published studies in that domain. Finally, we have a little bit of a distribution that you see when it comes to application areas where such studies are employed. So cognition makes up for 47 to 48% of the hyperscanning studies that are out there. And typically in these in this application area, you have a subject that is performing a task. So an audio paradigm, a visual paradigm, or a beat flash paradigm. So you are basically registering responses from a subject in that particular task. The next highest amount of study or application areas that exist for such studies is decision-making tasks. And this, for example, is when two subjects actually have to work with each other to come to a shared goal or a shared objective. So either they trust and cooperate with each other to come to that decision, or they use ways of deception and compete with each other to come to that decision. One good example is that study by Astolfi et al., where, for example, a pilot and a co-pilot are coming to a shared decision in a flight simulation, in a flight simulator. You see a lot of studies out there of the same kind. The third application area that you see a lot of these studies in is motor synchronization tasks. So that constitutes about 23% of the studies. And one good example there would be the Edson-Filio study, where they look into interactive juggling. So you have two sets of subjects. So you have individual juggling as well as paired juggling that they look into. They increase the difficulty of the task by increasing the number of things that they are juggling with. The other thing that also typically comes into this kind of application is when you're looking at a finger tapping task, for example. These are the three major areas which employ hyperscanning setups, but there's a significant percentage, so about 5%, that also look at educational context. And those, for example, are more in a classroom environment where you're looking at joint attention between the students, or you're looking at teacher to student relationships in the classroom environment. So this gives you a little bit of an overview of the application areas that we know for hyperscanning as of now. All of it can be found in the paper by Nam et al. This brings me to my next slide, because when you want to do EEG hyperscanning, there's some sort of architectures that are more popular in literature. This picture is taken from Babyloni et al. So what you see on as picture A on the left is by the paper from Babyloni et al. And what you see on side B is actually by Dumas et al. So side A actually shows you a little bit of a complex within the lab setup. You have 4 systems, EEG cabs connected to amplifiers with a trigger input. Now, and this is easily done in a lab where the distance between each system, between the 4 systems, is not too high. So you can actually have a physical trigger button connected to the amplifiers. There's an interface layer which receives the EEG data and the trigger information, and all of that is finally streamed out into an EEG workstation or a monitoring session. On the left, on the right, what you see is a more simplistic architecture. So you have 2 subjects, the data is recorded from them and kept in a system. So this could be, of course, streamed out in real time or stored in a network infrastructure for processing later on. These are more or less the 2 kind of general architectures that you can have for EEG hyperscanning. This brings me to my poll question number 3, because now, of course, as ANC Neuro, we want to focus a little bit on our range of EEG solutions, so eego. And my next question before I go to the slides is, are you curious about how the ecosystem can accommodate dyadic recordings? And here we've been a little bit smart with the options. So your options are yes, of course, yes, or I would like more info. I think Farnoosh would let me know once the polls are closed, and you have about 30 seconds to respond.
Of course, yes. So that's the answer.
That's great to know. Thanks, Farnoosh. So, of course, yes is a great response to move on to the next set of slides with. I hope with the next set of slides, I'm able to tell you how you can employ eego to achieve this. I would be covering 3 use cases. The first one is when you're looking at separate ego systems. Just to make sure that you see my screen. Okay. Now, the first application use case is where you have 2 separate ego systems. So there is a stimulus presentation PC, which has a parallel port, which can accommodate and send triggers from that parallel port to the ego amplifier simultaneously and synchronously if that is also demanded by the task in hand. On the ego side, of course, you have trigger adapter cables that we provide by default, which can pull in the hardware trigger to the amps. Each amp is connected then to a subject, and the subject wears a waveguard cap, as you can see in the schematic. In such a setup, you would need 2 recording devices. So subject data 1 is collected on recording device 1, subject data 2 is collected on recording device 2, and you have the triggers present in the EEG data to then synchronize the 2 data sets. This would be a relevant setup when you're looking at studies where subjects need to respond to tasks or stimuli. I do have a next slide on which tells you on what kind of hardware you need from our side to achieve this. And I would use the nomenclature that we generally use, but of course give you a little bit of explanation about the nomenclature. So first, being adapters, you can use the XS230 and or the XS231. The difference between the 2 is one has a DB25 connector, while the other one has a BNC connector. You could use an eego amplifier, and for the sake of consistency and to be more generic about what I cover today, we will be focusing on the 64 channel amplifier, which has the EE2XX nomenclature. Principally, this can very well be done by our 24 channel amplifier or our 8 channel amplifier, so the eego EE511 or the eego mini. You would need a waveguard cap from us to do this, and this would work for any sensor modality. So whether or not you're using our gel caps, our dry caps, or net caps, you would of course need a stimulus presentation PC, and this would have to have a presentation software. So either ePrime or PsychoPy, you could use anything that's out there. Finally, if you have a task which requires the subject to respond to the stimuli, you would need a response collecting device also. So this could be in the form of a push button, for example, or a haptic device that can register triggers and send it parallelly to the data sets. On the software side, you don't really have a very high requirement, so anything from eego version 1.8.2 and above would do. That's application use case number one, where you're looking at two separate ecosystems. We do have, of course, the need for hyperscanning in more mobile setups also. And this is what you should see in my current slides. So this is the kind of setup that we envision for when you're using eego in a mobile setup. You have a push button which can send a trigger simultaneously, so preferably which has a Y splitter, sends the trigger at the same time to amplifier 1 and 2. Again, this trigger can be sent at the start and at the end of the recordings. This does not need to be connected to the amplifiers at all times. Like the previous case, you have the data recorded then on recording device 1 and 2. What's an added measure is the eego sports backpack that we provide. Now with the sports backpack, you can actually load the recording device and the amplifier into the backpack. So all of it is on your subject. It's super lightweight. So our amplifier weighs about 500 grams and the tablet itself is not more than 1.5 kgs or so. And you have a relatively light equipment that the subject is carrying on their back. You also, with the backpack, leave the subject more free to interact with the environment, and they are not restrained when it comes to mobility. What we also have as a layer to make this recording easier is the eego web interface. So this is something that we will also employ to start and stop recordings across multiple devices, especially if it's in a mobile setup. As per always, I have a slide on what you need from us to achieve this. But before that, we want to look at what you would do to synchronize the data sets in such mobile setups. So we would recommend a TTL trigger, a photosensor, or similar for the first synchronization or device calibration. Again, you could use both TTL triggers and software events sent by the lab streaming layer protocol to send markers to your data. In each case, we would recommend timing tests done for that particular hardware setup, for that particular network infrastructure. This way you can take into account any sort of fixed offsets that you might expect in that network or with the hardware. Finally, all the event information with codes or the markers with annotations, with descriptions are actually visible on your EEG data sets. So you can actually visualize this on eego. All of the event information is, time included, is actually available to you during review also. And when you export the data finally from the eego platform, you also have this event information that you can extract from the CNT file, for example. Again, the most recommended way and the most foolproof way of doing this is, of course, the timing test first, but you would like to have a push button or a haptic device or similar so that you're working with hardware triggers instead of just a protocol that is more network dependent, for example. As always, I have a little bit on the hardware components that you need to achieve this. Primary being the trigger adapters, so the XS230 and the 231, like we said. So with the DB25 connector or the BNC connector, both possible. The DB25 connector actually has a direct input that can be on the amplifier. If you have a BNC adapter, you need to use it in series with the DB25 connector to actually then connect the trigger option that you have to the amplifier. We would recommend a push button with Y splitter in this case, because then it's easier to send in the hardware trigger at the start and stop of your recording. We would as always, say that you need a 64-channel amplifier at least, but you can also principally achieve it with lower channel counts on amplifiers. You would need the waveguide caps from us and eego sports backpacks in this case. In terms of software requirements, we would recommend that you stick with eego version 9.1 and above, because that is where the eego web controller feature was introduced, and eego web application is a nice added layer to have to make things simpler in such mobile setups. The final use case that I would like to introduce is number 3, where you're looking at a more integrated solution, or you're looking at a cascaded setup. And now this is relevant in a cognitive neuroscience lab, for example, where you expect both subjects to be stationary, and you can do this 2 ways. So what we see in the schematic is the eego hub from us, where you would just connect different subjects. So connectors A1 and A2 from subject 1 go on the top slot, connectors B1 and B2 from subject 2 go on the bottom slot. You have all of the data recorded from both subjects into one data set, which you see here on the right. And what Eego Hub also has is actually a parallel port. So the only thing we need is for the stimulus presentation PC to be able to send a hardware trigger to this Eego Hub port, or a DB25 port that is on the Eego Hub. For your purposes and depending on the task that you're running, you might want to have 2 screens attached. So if it's a visual paradigm, for example, you want to show subject A and B the same screen or the same schematic on the screen, and you have a keyboard or a mouse, etc., to register their responses. Again, this is very much possible also with the cascaded setup, which you don't see in the picture, but it is something that all of our amplifiers can do, especially the Eego MyLab amplifiers. Now, what you do in such cascaded setups is you have 2 64-channel amps from us. We use a cascading trigger adapter, and we use 1 bit to then synchronize these 2 64-channel amplifiers. With that, you have actually a 128 system that is cascaded, and you have then 7 bits possible or available to send in a TTL trigger through. So both these integrated solutions or cascaded setups allow you complete flexibility and allow you to actually record all the data from 2 different subjects or 4 different subjects into one data set. Finally, I have some sort of hardware requirements that you would need from us to achieve this. As always, we have these 2 trigger adapters that you saw that are available with an ecosystem by default, but you also have a cascading trigger adapter to, in case you're working with a cascaded setup, to basically synchronize 2 64-channel amplifiers. The waveguide caps, again, here is what you would is something that is up to choice. You could go with the gel or the dry. We leave that up to you. We do have to mention that the Eego Hub, as it is, does not hold as yet a stimulus presentation software. So you would require a separate third-party application that can generate the stimuli for your subjects. Finally, on the software side, again, as long as it's version 1.9.1 and above, it's not a problem to have synchronized data sets or one data set for all your subjects. This brings me to my next slide, because now we've covered the application schematic or the hardware components, but there's also on the software side maybe a question of how dyadic recordings will be accommodated by the Eego software. And here I think it's nice to mention that the Eego software, we both with our hardware and our software, we want to allow complete modularity and flexibility for the recordings and for subjects. And here on the software side, you actually could choose the device type or the amplifier type that is of interest to you. The software by default allows you to switch between various device types and amplifier setups. We have a workflow that is dedicated to creating or modifying amplifier setups. Amplifier setups, I should mention, is what you truly see on the hardware side. So for example, if a channel is checked off in this workflow, you do not record from it. We also have montages that are available by default or can be created, but there you really have the chance to play around. Now, what I say here is that you can modify an existing default setup for a hyperscanning experiment, and there you just decide how many subject sets you want to accommodate. So for example, you use the 256 default setup to record from 4 subjects, whereas you can use the 128 or the 64 default setup to record from 2 subjects. So for the 128 schematic, it would be more 64 channel data from 2 subjects, whereas if you were recording on 64 channel amplifiers or 64 channel setups, you can actually use it for 32 channel recordings from 2 subjects. Like I say here, you have to edit one of the default setups to accommodate 2 subjects or 4 subjects data. You can always take a look at the CAP data sheets for the pinning or the order of channels. I have a little bit of a schematic here because I think a little bit more visual input helps always to understand. And here I would repeat what we said already in the beginning, that you have these couple of combinations that you can do. So you have 2 subjects, for example. So we're talking about diegetic recordings, and the data from these 2 subjects can be recorded with and on 2 different amplifiers and 2 different recording devices, or you record both their data as one cascaded setup and one recording device, so you have it all in the same data file. The next layer is that depending on whether or not you have a cascaded setup or you have 2 separate 64 channel amps, all your data actually goes into one recording device, or all your data goes into 2 separate recording devices. And that decides what kind of setup you use, of course. So if you are looking at separate 64 channel devices and amplifiers, you can have the default 64 channel setups. Whereas if you're looking at a cascaded setup and one recording devices, like I said, you can look into a default 128, 64 or 256 channel setup. Finally, what you do on the software, right? You have this workflow. So we have 2 of these administration workflows, one being the edit amplifier workflow. Now here you can actually adapt or copy from a default setup to be ready for a hyperscanning setup. What you would need here to do is to check the CAP datasheet, which is provided with any waveguard CAP that you buy from us. It gives you the pinning of the channels and the order of the channels. So in the same order, you would adapt the 128 default setup on the amplifier, for example, for subject 1 and 2. So you would have the same 64 channel sets for 2 subjects, repeated one after the other and contained in a 128 channel setup. In the edit montage workflow, you really can play around with visualization and filters and to see how you want to differentiate between data from subject 1 and subject 2. Again, this is what you do with the edit montage workflow is visualization only. But it might be easier for your purposes or for the EEG experimenter if you say that you want to see all of subject 1 data in color blue, all of subject 2 data in color red. All of those options you can explore using the edit montage workflow. Essentially on eego, you need to edit the amplifier workflow, you need to edit the montage workflow. Both of these can be copied over from existing templates, so which makes the users or the researchers work significantly easier. With that done, you're actually very ready to go. So you have a subject created, of course, you can call it whatever you like. You just need a first name and a last name, which could also be identifiers. And you need to start with the acquire workflow or the acquisition workflow, and you would be able to record data from both subjects into either one data set or separate data sets. This is how you would do it on eego, more or less. And so as you can see, it's pretty easy done. It does not come with added complexity. I think it's nice to go over some use cases that have been phenomenal and recently out in the field. It gives you a lot of incentives or encouragement to try out these hyperscanning setups yourself. The one exemplary use case that I must mention here is the NeuroLife project. We have a showcase coming out on this soon, coordinated by one of my colleagues. But this study was an interdisciplinary team or research endeavor by Goldsmiths University of London. This is it's the first one-of-a-kind multi-system, multi-device EEG hyperscanning study that looks into investigating liveness and audience experiences. What they do is they use 21 32-channel eego sports systems in parallel. You actually see here that the audience members are wearing our equipment. They also have a respiration sensor from us attached to them. And what they're doing is they're watching the performance that you see on the left-hand side, which is called detective work. So it basically is a choreography which was coordinated by the Siobhan team. And this is actually one of the largest install bases out there that we have that have tried also the eego web controller and LSL functionalities simultaneously. I have to mention that they use a photodiode at the beginning of the recording to synchronize all devices, so all 21 devices. And then during the recording, they had an LSL script in place that would send markers at regular intervals to the EEG dataset. They use the eego web controller, start and stop recording across 21 devices at the same time. That is the NeuroLife project. We are very excited to see outcomes of the research. Right now, there's very less material published on this, but we hope to see something more soon. The second use case that we want to highlight is the Filio and David Stone paper, which investigated cooperative juggling. What they did is that they used 32 eego sports systems on 2 jugglers of varying skill levels. So varying skill levels, I mean, so either more than 3 years of experience or in the line of or under 3 years of experience. They ran both individual juggling and paired juggling tasks with both these skill matched or skill unmatched jugglers, and the outcome shows that in between individual and interactive juggling, there's distinct patterns of functional connectivity. And these differences that they saw were most dependent on skill level. The reduced functional connectivity that was noticed during interactive juggling also goes to show that it is most likely harder than performing individual juggling. There's a lot of interesting outcomes that are part of the paper, and we do suggest that you take a look to go over all of the analyses that was done by the group in this area of interactive and paired juggling. The next exemplary use case I have here is a recently published study by some of our researchers in University of Kent. Rosso et al., for example, looked into simultaneous EEG recordings between 2 subjects while they were performing a finger tapping task. They employed 2 64-channel eego systems in the study, and what you see here is basically, so they have a carrier frequency, which is more a high frequency component, so about 20 hertz, and they have a modulator frequency, which is at the same frequency of the tapping task. And then they have these self and other graphs that you see towards the end, and what they finally show as outcome is that periodic beta modulations actually played a good role in enabling adaptation and cooperation mechanisms between coupled individuals. They also found that the beta power was modulated to a greater extent by self-produced finger taps as compared to other produced finger taps. It's one of the recently published studies in the realm, and lends insight into such interpersonal synchrony between dietic couples or recordings. The final use case that I have here actually is not is more a review article than a study itself, but I thought it was a good idea to include it because this lends an insight into kind of on the variety of application areas where you could adopt hyperscanning studies. Now, this is a picture from the researchers or the group led by Francisco Parada, where they really want to focus on bringing naturalistic settings to the lab or running more ecologically valid setups. Now, with the equipment and the tech infrastructure that we have now, you could actually extend natural settings or naturalistic recording settings to fields such as psychotherapy, architecture and design, for example, and neuroprosthetics. This is just something to keep in mind for researchers that are looking at really pushing the boundaries of what exists out there and how much they can do with the current tech infrastructure that we have in place. This actually brings me to an idea or a slide on the methodologies that are out there for EEG hyperscanning analyses. Here you see that there are 3 articles that I have listed up. We do want to share a word of caution that all of these approaches are specific to the setup that is looked into within the studies. Now, a lot of these studies where cooperation or connectivity is being explored look at a phase locking value, for example. So if the phase is locked within a certain time window, the value is 1, and then you have more synchrony in the phases between two subjects' data. And if the phase locking value is 0, it means that there is no phase synchrony between the two subjects' data. Another application or another method that they do employ when it comes to finding out more information about, especially in the area of decision-making tasks, is graph theory. And graph theory basically gives you an idea about brain-to-brain links, let's say. And for those kind of studies, graph theory is something that is employed very much. The third kind of analysis method that is used is partial direct coherence, and this gives you an idea on the flow of information between brain 1 and brain 2, so between subjects. And this kind of analysis method was also employed by Astolfi et al. In their paper, which was to do with two pilots cooperating with each other in a flight simulator. So this is just an idea of what analysis methods could look like for hyperscanning, but we would suggest that you take a look at these review articles, which give you the whole overview of what you could adopt for your particular study type. I do have a slide on what exists in terms of toolboxes and data sets out there, because a lot of us are new to hyperscanning analysis, and it's not so easy done as in regular EEG data, right? Because we're looking at a lot of variables, and we're looking at inter-brain synchrony in a lot of these cases. So the first two are actually toolboxes that exist. So DEEP is a dual EEG pipeline for developmental hyperscanning studies. And what they do is they have a pipeline that basically imports the data. They use an ICA decomposition to remove any artifacts. Then they actually look at phase synchronous values, and they perform a power analysis. Finally, all of the analysis is actually averaged over the diodes. It is available and easy for you to use if you want to take a look. The second is actually HyPipe, which is a hyperscanning Python pipeline for inter-brain connectivity analysis. Again, here what they do is they import the raw data. They have a pre-processing box or step. Finally, you have either a statistical analysis or a visualization of the data between the two brains. You would need Python version 3.7 or above to run this, and you would need usual libraries for plotting such as Matplotlib and Pandas to make use of this toolbox. There's also databases that are out there that cover dyadic recordings or multiple recordings from diodes. One is test code. It's basically that has more sort of social sort of interactions recorded, so gestures and smiles, for example, and you can use this to see what dyadic interaction analysis might look like. The next is the NOXI database, where they have different sorts of inputs. So they have motion capture data, they have gestures and smiles again, and you have all of this data in novice expert interactions. Again, the database is freely available if you want to look at a little bit of dyadic hyperscanning data or dyadic data that you could employ to find out more about what you do in a hyperscanning context. This actually brings me to the slide on challenges. Like I said, as with any realm, we have challenges when it comes to hyperscanning studies too. The first is that when you have a structured or semi-structured session, it is actually easier to control the experiment. So in a typical lab setting, for example, or in a stationary setting where the experimenter can really control the environment variables. But the idea to have hyperscanning studies is also to make sure that it is a more ecologically valid or naturalistic setting. And for those kind of setups, it is important to allow the subject free interaction with the external environment and or other subjects. So we recommend that that another modality is also recorded from as a sanity check or a measure. In fact, this is already being done in a lot of studies that were out there. So they have like eye tracker information, they have EEG information, or they have motion capture data as an added measure, especially in mobile context, for example. This brings me to my next slide, which is more on unstructured setups. like I said, the goal is to bring naturalistic settings to the lab, but that is harder said than done. Secondly, the community is very, very new to handling hyperscanning data. It is still what we would call a young science. And this poses its own challenges because there's no gold standards that exist in the case of data analysis. It remains more study dependent. Finally, when we have multiple devices that are to be synchronized and calibrated before they are run in parallel, we do recommend that a general testing is done. It is in fact critical and should be done prior to the pilots being run, because this is the only way you can take into account fixed offsets that can be part of your hardware, that can be part of your network infrastructure. Finally, hardware triggers or photodiodes are what we recommend to really use for device calibration and synchronization. So as long as we are aware of the challenges and we're ready to go down the path, I think it is still a worthwhile concept and a very useful topic to really explore within your analysis and research. What we will have next is the demo video, but before we go to it, I would actually want to leave a question out to the audience, and that being, what other applications could you envision with the hyperscanning setup? I did my best to sort of cover a couple of research and application use cases that are out there, but maybe you have more to share with us, and we look forward to knowing what your ideas are on it. With that, I actually am at the end of my presentation.